Yeah, I've started.
So, a very good evening to everyone present here. We welcome our moderators for the session today, Dr. Sushma, Assistant Professor, Department of Pharmacology, and Dr. Prashant, Assistant Professor in Department of Physiology. Uh, Today we have Rohan presenting a journal club meet on the efficacy and safety analysis of prucalopride in refractory chronic constipation cases in a tertiary care hospital in eastern India. It's a randomized single-blind placebo-controlled study. Uh, yeah, Rohan, please do begin. And this is our first journal club meeting ever since the new core team has been founded. Really excited to have a great year with you all.
Thank you, Manini. I'll be presenting my screen now. Hope I'm audible.
Yes, you
Yeah.
are, Rohan.
So, good evening, everyone. Hope the slide is visible.
Yeah, Rohan.
Yeah. So, good evening, everyone. I am Rohan Nikhil Dabir from second year MBBS, Bangalore Medical College and Research Institute. Here, I'm presenting a journal club on efficacy and safety analysis of prucalopride in refractive chronic constipation cases in a tertiary care hospital in eastern India, a randomized single-blind placebo-controlled study. So let's first talk about which journal was it published. It is published in Indian Journal of Pharmacology. When we come to the journals, we have something called as a journal impact factor. So the journal impact factor is calculated by dividing the total number of citations in the previous 2 years by the number of source articles published the following year. So if we see the impact factor of Indian Journal of Pharmacology, it is 2.833. We have some journals which are highly rated through this impact factor. They are the Journal of American Medical Association, the Lancet, and the Lancet Respiratory Medicine. These are very highly rated journal with high impact factors. So let's come to authors. As we can see, all authors are from the Department of either Pharmacology or Department of Gastroenterology from the same college or hospital. So what we can comment on this is there are no guest authors because no one is from the journal or anything, and there are no ghost authors because we can see that there is no high influential person working for their article. So grossly, the abstract is well structured and it matches the text of the article. The keywords are also very appropriately given. So chronic constipation is a common problem and the main treatment is usually laxatives. However, some people do not respond well to laxatives. So researchers wanted to see if new medication called prucalopride can help. They conducted a study with 180 adults. This conducted a study with 180 adults who had constipation that didn't respond well to laxatives. The patients were randomly divided into groups, one group taking prucalopride and the other group taking a placebo pill. The study lasted 12 weeks, during which time the researchers measured the number of spontaneous bowel movements the patient had each week, as well as other factors such as patient satisfaction with treatment and changes in the stool consistency. They also monitored the patients for any side effects or changes in heart functions or blood tests. The results showed that the prucalopride was significantly more effective than the placebo in increasing the number of spontaneous bowel movements each week. Patients taking prucalopride were 41% more likely to have three or more bowel movements per week compared to those taking the placebo. In addition, the prucalopride group reported greater satisfaction with the treatment and improvements in their constipation symptoms compared to the placebo group. The common side effects reported were headache, nausea, bloating, and diarrhea. But there were no significant changes in heart rate function or blood tests. Overall, the study suggested that prucalopride is a safe and effective option for people with chronic constipation who do not respond well to laxatives. So, let's begin with the research paper in detail. Chronic constipation is a widespread issue that is typically treated with laxatives. However, some individuals do not respond favorably to the laxatives. So, what is a true refractory constipation? A chronic constipation can be labeled to be a case of true refractive constipation if, after careful diagnostic and therapeutic evaluation, he or she fulfills standard diagnostic criteria for functional constipation, lacks any alarming feature of organic conditions, and fails to improve upon intake of high fiber diet and laxatives, either a stimulant or osmotic ones. So, although many drugs are available in the market, pharmacotherapy of chronic constipation still poses a great challenge. Because of wide patient dissatisfaction and increasing refractiveness, there is a need for effective treatment for chronic constipation. So this led to the discovery of a new class of drugs called prokinetics. They increase intestinal motility, giving relief to the constipation symptoms. Cispride and tegaserod are now withdrawn due to dangerous cardiovascular side effects. So cispride and tegaserod belong to the class of drugs called prokinetics. They are 5-hydroxytryptamine 4 agonists. So they are banned due to they have dangerous cardiovascular or cardiovascular side effects. So, because of this, there was the discovery of prucalopride, which was recently approved by Food and Drug Administration in 2018. This is also a 5-HT4 receptor agonistic activity. This also enhances colonic motility. So, here we are trying to check how good is this. Should that be used for treating chronic constipation? So the study is a prospective, randomized, single-blind, placebo-controlled, and a parallel group study. So let's go through it carefully. What is a prospective study? A research study that follows over time group of individuals who are alike in many ways, but differ a certain characteristics, and compare them for particular outcome. For example, female nurses who smoke and those who do not smoke, and the outcome measured can be lung cancer. So this is a prospective study. Then a randomized study is a study in which participants are divided by chance into separate groups that compare different treatments or other interventions. Using the chance to divide people into groups means that the group will be similar and the effects of the treatment they receive can be compared more fairly. At the time of trial, it is known which treatment is the best. So the next it is single blind, a type of clinical trial in which only the researcher doing the study knows which treatment or intervention the participant is receiving until the trial is over. A single blind study makes the result of the study less likely to be biased. This means that the results are less likely to be affected by the factors that are not related to the treatment or the interventions being tested. So the next is placebo control. It refers to a clinical study in which patients receive a placebo. So what's a placebo mean? Placebo is an inactive substance or any other intervention that looks same as the given testing drug or the active drug. The effects of the active drug or the other interventions are compared to the effects of placebo. So the next is parallel group study. The opposite of parallel group study is a crossover study. As I told you in the introduction, uh, some population was given prucalopride, then other population was given placebo, and for the whole follow-up of 12 weeks, they were just given the group A was given prucalopride, group B was given placebo. That's a parallel group study. In a crossover group study, what is done is initially they give placebo and prucalopride respectively, then they exchange them. This is called a crossover study. But the research paper which we are studying now is a parallel group study. So now, here a question comes. What is the rationale for a placebo study? So as we can see, it was approved. The prucalopride drug was approved in US and Europe long ago. It was approved in India by 2018 and was allowed for using in therapeutically. Then why was the need for doing a placebo study? This question is posed to the research paper. So the next is, the study was conducted in collaboration with the Department of Gastroenterology and Department of Pharmacology in the SCBMCH Cuttack, Odisha. So this study was conducted for the duration of 12 months and prior ethical approval from the Institutional Ethics Committee of that college and written informed consent from the study subjects were obtained before the study began, which is a positive point and a very important and necessary for all researchers to be undertaken. So the next is Clinical Trial Registry of India. ICMR has organized a clinical trial registry of India called CTRI, but this clinical trial wasn't registered. This is a negative point which we can see in this research paper. Every clinical trial should be registered in the CTRI. So, what advantages does it pose if we register in the CTRI? So, the registration of trials will ensure transparency, accountability, and accessibility of clinical trials. It prevents selective reporting and publication of positive results. It prevents unnecessary duplication of research efforts. It empowers patients and public about planned or ongoing trials and, if necessary, enroll them. It gives ethics committee and researchers across access to studies being conducted and compare them to the research that they are considering for or being planned. So patients who are at least 18 years of age of either sex and presented to the outpatient department of gastroenterology with a history of chronic constipation, fulfilling the Rome 4 criterion and the non-responsiveness to the laxative treatment for the last 6 months were included in the study. The question here comes is which laxatives? They have not mentioned clearly which laxatives or non-responsiveness to which laxatives, which shows there's a lacuna in the inclusion criteria. There's also no mention of what is responsiveness, non-responsiveness. The paper hasn't defined any sort of criteria where non-responsiveness for 6 months, but what dosage of laxatives is not mentioned. There's again a lacuna in inclusion criteria. Then, patients with secondary causes of chronic constipation or any cardiovascular, liver, renal, or psychiatric disorders or any known adverse drug reaction to study drug and excipients, pregnant and breastfeeding women were excluded. As I told you, the previous two drugs which were cancelled due to cardiovascular risk, so patients having cardiovascular risks were excluded. Then liver and renal is very common to all drugs. And then what's the reason for psychiatric disorders? It was also observed in the previous two drugs that people taking had psychological changes and had there was increased suicidal attempts in these patients. That's the reason even people suffering from psychiatric disorders were excluded from the study. So out of 560 patients screened, 180 patients who fulfilled the inclusion and exclusion criteria were randomly allocated to two groups using the random table. How was randomization done? The article says that the patient who came to the table was randomly allocated by the doctor to group A or group B. There's a mistake or there's a lacuna over here. It's randomization should be in a closed envelope that neither the doctor nor the patient should be knowing who is which treatment is being given. That's called a closed envelope blinding, which is not being followed over here. So group A, there were 90 patients to receive tablet prucalopride 2 mg, and group B, again 90 patients, were to receive placebo. As I told, it's a similar looking tablet. They were given it once daily just before breakfast for a period of 12 weeks. Follow-up was done at 4, 8, and 12 weeks. So there's a good point to be noticed here that no patient withdrew from the test or no patient missed the follow-up. This is very rare that the patient comes for follow-up every time. Like among 90 plus 90 patients, that are 180 patients, that all 180 patients came for the follow-up or no one withdrew from the study. This makes us doubt regarding the research paper. So, in this study, who did not have bowel movements for 72 hours during the 12-week study were given a rescue medication, specifically a 10 mg biscodile tablet twice a day. If the patient didn't respond to the rescue medication also, they were given enema. The study tracked how many rescue medication each patient used during the study period. However, 48 hours before and during randomization, rescue medication and enema were not allowed. So the primary efficacy endpoint was the percentage of the patients having on average three or more than three spontaneous complete bowel movements per week, the number of SCBMs, that is spontaneous bowel complete bowel movements per week, and the number of spontaneous bowel movements per week during the 12 weeks of the study period. Secondary efficacy points included the assessment of improvement in chronic constipation symptoms using validated 12-item patient assessment of chronic constipation symptoms. This is called PACSYM questionnaire. And it is subsymptom score which included abdominal, rectal, and stool symptoms. The overall PACSYM score was anywhere between 0, that is absent, to 4, very severe. Assessment of stool form was done using Bristol Stool Form Scale through 4, 8, and 12 weeks. Overall treatment satisfaction assessment was done using a 5-point Likert scale at baseline at the end of 12 weeks. Safety data of all participants who took prucalopride even once was recorded. Vital signs along with the ECG findings were recorded at the baseline and at the end of 12 weeks. Any ADR reported during the study duration was noted in the suspected ADR reporting form of Indian Pharmacopoeia Commission, National Coordinating Center, IPC NCC. Casualty assessment and severity assessment were done using WHO UMC scale and Hartwig's severity scale, respectively. So let's come to results. In this hospital based study, the researchers recruited patients who had chronic constipation and had not responded to laxative treatment. Patients were randomly assigned to receive either prucalopride or placebo for 12 weeks. The study found that prucalopride was significantly more effective than placebo in increasing the number of spontaneous complete bowel movements per week, as well as in improving secondary endpoints such as the number of bowel movements with normal consistency, the number of bowel movements with the Bristol stools scores 4 or 5, the reduction in the use of rescue medication. The study also found that the patients in the prucalopride group reported their first SCBM earlier than those in the placebo group. The study included patients who had mean age of 59.8 years with a range of 30 to 70 years, and the most of whom were males, 59%. The patient had history of constipation for an average of 6.7 years, and the most common comorbidities were peptic ulcer disease and anxiety. Prior to the study entry, the majority of the patient expressed dissatisfaction with their previous treatments with laxatives. The study found that at baseline there was no difference in the number of SCBMs per week in prucalopride and placebo groups. However, after 12 weeks of treatment, a statistically significant number of patients in the prucalopride group had a higher number, or 3 or more SCBMs per week, than in the placebo group. But what we can observe here, just look at the graph properly. I'll explain it in the next slide. So the mean number of SCBMs over 4 to 12 weeks were also improved in the prucalopride group compared to the placebo group. But there's a problem over here. The number of patients with average of more than 3 SCBMs per week was taken just at baseline and just at 12 weeks. The subsequent parameters like number of patients with an average increase in bowel movements was taken at week 4 and week 12. The number of patients with an average increase of greater than 1 SCBMs per week was taken in week 4 and week 12. But in the previous graph, we can see they've plotted data for the 4th week and 8th week also. Their graphs, their tables show that they have not taken any follow-ups during the 4th week and 8th week, but their graphs show that they've taken data from there also. There's a small lacuna over here. What I suspect is they might have taken the 4th week and the 12th week and just joined the 2 points as called extrapolation. So the study found that the baseline there was no difference in the SCBMs per week in prucalopride and placebo groups. However, 12 weeks of treatment, a statistically significant increase in number of patients in prucalopride group had higher number, or 3 or more SCBMs per week than in placebo group. The study also found that the patients in prucalopride group reported their first SCBMs after 2 days of treatment initiation, while patients in the placebo group reported the same after 5 days. The use of rescue medication was also reduced in prucalopride group compared to the placebo group. So here we can see the ADR distribution also. So, chronic constipation is a multi-symptom complex disorder that has a major impact on physical, social, and mental well-being of subjects. It affects persons of all ages and sex with a very negative effect on the quality of life of patients in the long run. With increasing refractoriness to the traditional laxatives, treatment of the same has become cumbersome. The study found that the prucalopride was effective in relieving symptoms of chronic constipation by improving impaired colonic motility with a lesser side effect profile. The test group patients achieved significantly higher percentage of changes in the primary efficacy endpoint of 3 or more spontaneous bowel movements than placebo. Other secondary efficacy endpoints like improvement and relief in constipation symptoms validated via the patient assessment constipation symptoms questionnaire at the baseline 4, 8, and 12 weeks were significantly high in prucalopride arm than in placebo arm. The study was conducted in an Indian population comprising adults with refractory CC and had a male predominance of 59%, which was in line with the study conducted by Ray and Shah et al., which is one of the references. However, its findings in contradiction to several Western studies where female predominance was seen. Peptic ulcer disease and anxiety were the most commonly found comorbidities along with CC in this study. The response rate among patients when followed through 4 to 8 weeks were highly significant in the prucalopride arm than placebo. Patients in the prucalopride arm through 8 to 12 weeks reported less use of rescue medication than placebo. Treatment satisfaction among patients were higher in prucalopride arm than in placebo. Prucalopride was generally well tolerated for 12 weeks of the study period. Adverse events recorded were mostly GI symptoms like mild to moderate in severity within known pharmacological side effects profile of the drug. No new safety signal was detected during the study period, or no clinically relevant abnormal cardiovascular parameter with therapeutic dose of prucalopride was reported. Limitation of the study included a small sample size with a single centric approach and a duration of only 12 weeks. Further trials with different doses of prucalopride on Indian population are required to establish a high margin efficacy and safety profile. Reading the discussion, we understand that they have just copy pasted and well written part of the results. We cannot see anything new explained in the discussion except the limitations. So, in the near future, as more people in India adopt westernized lifestyles and make changes in their diets, there will be increasing number of people who do not respond well to the standard treatments for constipation. This would have negative effects on their quality of life and productivity. This study, which is the first of its kind in India, suggests that prucalopride taken once daily along with changes in diet and lifestyle may be a viable treatment option for treatment for patients with chronic constipation who have not responded well to other treatments. However, more extensive clinical trials involving various types of constipation patients are necessary to determine the long-term effects of prucalopride. These trials should include patients for all ages, including children, the elderly, and those with constipation caused by medication or motility disorders. So, in conclusion, what I can say is this research first thing, there was no need of a placebo controlled research as the drug was well established. Number two, they have just concentrated on a 12-week follow up, which many other research papers in from UK or US have been done. They should have concentrated in long term effects of cardiovascular system or maybe even psychiatry in that case for this drug of prucalopride. They have just concentrated on the 12-week repeated research which has been done in many other centers in many different types of setups. So this was the journal club on pharmacology. Thank you.
Thank you, Rohan. That was a really great presentation, especially the PPT. It was really, really nice. So, yeah, does anybody have anything to add and say? And definitely our moderators.
Ron, I think on the sample size, you want to give a comment on the sample size?
Yes, ma'am. The sample size calculation was not done according to the sample size formula. It was just randomly taken. If proper sample size calculation would have done, it would come 180 in per group. That is total 180 plus 180 per group. But what they have taken is 180 total and 90 plus 90. One more mistake which we can point out.
Can you comment a little bit on the tables also? Just put up the tables back,
Okay.
starting from the baseline. Okay, so
Yes,
whenever we put up a baseline characteristics in this table, what do you want to see and what is not there?
first thing, they haven't mentioned the software which they've used in doing the data analysis, and they haven't even mentioned the p-values for each parameter they've used, like whether it's significant or non-significant. They haven't mentioned that in the table.
Okay, see, for everybody, I just wanted all of you to know whenever we put up baseline data, you want to say that both the groups are comparable. So we want you to put up a p-value, and then every table is called a standalone table. So in standalone tables, we want the statistics, whatever they have used, it can be t-test or chi-square test is used, and significance is p less than 0.005. At least that much we need on the table. And then we don't have a p-value here, but the text says that both the groups were comparable due to randomization. Okay, and the next table, the primary endpoints, and this figure too, I just want all of you to know that sometimes people, you when you are publishing initially, you think the tables are not looking good in my article, so I have to put up one graph or a figure and put up a figure like this. Okay, so as Rohan said, they might have just extrapolated, but that may not be the case also. They might have calculated, we don't know, but they have somehow connected the dots. But for the placebo, we don't see, you have to sort of guess what is the number. And here again, we don't know what is the statistics they used. And there has to be some amount of repeated measures ANOVA to say that even prucalopride worked over the weeks and things like that, which is again missing here. And again, next table, Rohan. The primary efficacy points, again, what you can see is when the follow-up is from baseline to 4 weeks to 8 weeks to 12 weeks, you present the whole data to the audience. You just don't select things which looks good on your table. So that was the comment. And then again, statistics there, as I told you, they're called standalone tables. So if I give you one table, you should be able to tell me what is the whole article about. Okay, so your table should be that good and that informative. So these tables lack what is the statistical test used, what is the significance, and again, as we see, sometimes it is 4 and 8 weeks, sometimes it is 4 and 12 weeks. So those kind of data you cannot present. Always you're going to present everything to the audience, not selectively report things. Okay, so these were about the tables. And again, as Rohan said, from primary efficacy points, they kept saying that the patient will maintain a dairy, but we don't know how they calculated how many movements were there. They said average movements over 12 weeks. So you are going to add all the number of movements and divide it by 12. So those kind of little things, when they don't give, then everybody gets a doubt, how did they calculate and arrive at this number. So those kind of details are very, very necessary. And as Rohan said, they have not given us how they are going to compensate for the missing data. And luckily for them, R180 continued in the trial, which is very, very doubtful. Okay, first of all, you may think they are going to continue, but still you should consider that 10% missing data and how you are going to do the calculate the missing data. They are called as imputational methods. You guys can sort of read it. What are different types of imputational methods? So how do you compensate for the missing data? That is one. And then what else? I think we had a comment on the title as well.
Yes, I'll just bring the title.
Yeah. Yeah, you can tell what is wrong with the title and how you wanted your title. The title
Yes.
of the article should be.
So the title of the article, what they have given is Efficacy and Safety Analysis of Prucalopride in Refractive Chronic Constipation Cases. The cases looks odd, so the better word would be patients. it would be more human to be put properly. The efficacy and safety analysis of prucalopride in refractory chronic constipation patients in a tertiary care hospital in eastern India, a randomized single-blind placebo-controlled study. The positive point to be told is they have mentioned the region where they're doing the test, and they're telling even the what type of hospital they're doing the test, and exactly they've mentioned what kind of studies.
So calling people as cases and all that, even when you become PGs and you start writing your synopsis, that is a very common mistake we see. So don't just call anybody as a case and all that. So you'll say patients with refractory chronic constipation. Yeah, and then as Rohan mentioned at the end, so at the end of journal club, only one question we all need to ask ourselves is this based on this study, am I going to change my clinical practice? That is the first take home message. So based on this study, are we going to change our clinical practice? Here, Rohan's answer was no, because one, we already have a placebo controlled trial. Okay, so they kept saying that you have to give it in refractory constipation, but we went back and read the Indian consensus guidelines on constipation. Prucalopride is already placed in the guidelines from 2018 itself. So they have said if the patient has a proper, they do this physiological tests and say whether the patient has colonic transit time is correct. If they have it, then prucalopride is definitely given. If prucalopride doesn't work, then they are going to give rifaximin. So in 2018 itself, Indian guidelines already has prucalopride in it. So we don't really require placebo controlled study. So this result is just an addition to whatever was already there, and that is why we said that it is waste of resources that you do study like this. But we want to know, because prucalopride is a new addition, we want to know in long term how the patients are responding to it. Maybe till 12 weeks in all clinical trials. Apparently there are 50 plus clinical trials in prucalopride with over 6,000 patients. Clinical trials have been done all over the world, so this was not required. But what we really need is what happens after 12 weeks. Do they really continue to respond? For how long do they respond? And as Rohan mentioned, what is the safety data over a year or two, or when they become non-responders, and what are the factors which makes them a non-responder? So if we put our funds and resources towards finding answers for that, then it is a good research. Otherwise, this kind of repetitive research definitely should be stopped when the guideline itself has prucalopride, the two Indian guidelines. So that is what we had. Rohan has anything else to say?
No, ma'am. I think we have covered everything.
Yeah, sort of. We have covered everything, and I think any other points for discussion we can take.
Ma'am, like Prashant sir, if he can add some points. Right?
Yeah. And anybody, even I think all the other students also can ask a question or.
Ma'am, would you comment on the blinding of the study? Like, why do you think it was essential, and what kind of blinding is useful for which study types?
Okay, see, blinding, I think apparently now we are not supposed to say the word blinding. We should say it as masking. Yeah, so that is okay, but I think we are still accepting. I think all of you just read about what is single blind, double blind, and triple blind. So whenever we say blinding, always triple blind is your patient doesn't know, the doctor, investigator who's doing the study doesn't know which which patient is receiving what. And ultimately, the person who is analyzing the data also doesn't know, because even for him, we have blinded everything. So that is a very good thing which we have to, if we consider doing RCT, triple blinding will be very, very good. But it completely depends on what sort of resources we have, finances we have, and technical help we have. So even single blind, if your patient doesn't know what he's taking, is okay. But in this study, what happened was randomization table itself was given to the physician who was treating, and then he decided based on the number, randomization number he got, and which patient went to which group. So what best we can do is you are going to generate a randomization sequence. So if you put in number, there is a randomization.org and all that. So if you put in how many patients you want to include in the trial, like 30 patients I want, there are groups A and B. Then it is going to generate a sequence A, then B, then A, B, like that. Okay, different sequences it gives. So you can say patient number 1 is A, and then close it. And whenever you make different envelopes, putting all these 30 into with opaque envelopes where I can't hold it to the light and see which is next group. So, and you hand over everything to the investigator, and he's going to properly take out those opaque envelopes and open it and see the group he belongs to, and then give him that treatment. At least that we should do, which is called as allocation concealment. See, the thing is, if I'm only sort of generating a sequence and deciding which, if I know that the next patient is going to be, and I sort of know B is placebo, and my friend comes in as a patient, then I'll tell him, you just go have coffee for an hour, eat something, and then come back. So that one patient comes, I give him placebo, and my friend gets an active drug. Okay, so those kind of things happen if you don't do allocation concealment. So all of you have to know the difference between what is blinding and what is allocation concealment. And then, as you know, we take a lot of RCTs into meta-analysis. Meta-analysis takes very well performed RCTs. So how do they know your RCTs are well performed? They do something called as risk of bias. In risk of bias, this randomization, blinding, allocation concealment, all that determines the quality of your RCT. So if you want to generate good research data, you have to give importance to all these points. I hope I am clear. I think little bit of reading should be done by you guys also.
Yes, ma'am.
Yes, ma'am. Thank you so much. Ma'am, also, is there a difference between how we should approach a journal club meet for an RCT versus any other paper or a meta-analysis or a systematic review or something?
Yeah. Mani, that's a very good question. I think I asked Rohan to put off the concert checklist as pop-ups. I think he was scared about the time. So it was taking a lot of time. So whenever there is RCT, you have concert checklist. I think I put it in science club. And then whenever there is observational studies, you have to follow the stroke checklist. So what we tell our PGs is, once an RCT is done, again you put up the concert checklist and start answering all the questions. Like scientific background is well explained in introduction. Then I'll say yes or no as my comment. So all our any student who's doing a journal club have to put up the concert checklist. No, I mean, if you after introduction you want to put up the questions, you can put up. But if you don't want that flow to go, then you can present your journal club, and at the end you can put off the concert checklist as a whole and say what is given and what is not given. Now this concert checklist has to be followed even for publication. Okay, so all the journals, you have to, if you are presenting RCT to a journal, you're sending one of your RCT articles to a journal, all the questions in the consort checklist should be answered in your article. Okay, and for observational, it is strobe checklist. And if you're doing a meta-analysis, it is Prisma. And if it is diagnostic, it is STAR D guidelines. So I think we can start taking next to observational, and then after a few, we can take a meta-analysis also, and then we have to follow those guidelines.
Thank you so much, ma'am. Thank you for that. Prashant sir has been raising his hand. Is there anything, sir?
Yeah, congratulations, Rohan, for that excellent presentation. And I must also say the PPT is like next level technology. It made for a very pleasant viewing and also made it very clear. Congrats on
Thank you,
that.
sir.
And a lot of things related to the pharmacology class, and generally in research methodology has been told by Sushma. I just wanted to add a few things. Generally, for everybody who will present journal clubs, at least when you start presenting it, one thing is, as ma'am said, in the abstract you will anyway present the overall picture of the study, right? After that, I think it makes sense each item you critique on it, so that like when you present their introduction, you say whether the study is adequately justified by the authors, or whether the knowledge gaps have been adequately identified, and whether what is the benefit that is expected out of this study, whether it could just be merely addition to knowledge or change practice guidelines or whatever, whether it's been justified or not. So that you said this, in the end we saw that it really did not contribute all that much, so that part. And also, particularly for an intervention or a comparative study, it is better to hypothesize whether they have hypothesized or not. And if the authors have not, I think it is a good practice for you presenters to try to formulate a hypothesis. So that will help you when you design your studies and when you write up your articles and all that. So that is one thing. And I don't know if I missed it or whether the objectives were stated at all or not. I'm not. I didn't. I don't remember seeing a slide where whether the authors have mentioned the study objectives or not. If they have not, again, if they have, you should also again say whether the objectives are specific, measurable. Those things you have to comment about the objectives, and it is a very good practice for you also to start formulating your objectives well, because the rest of your study will hinge on very well formulated objectives. So that is one thing I wanted to tell. And sample size, yeah, you said the rationale has not been explained. One thing, and one one more thing. Um, it is also like you very nicely explained the study design, meaning why is it called single blinded, why is it called placebo controlled, all those components you explain, and why this particular study design is appropriate to these objectives. Likewise, at least when you begin presenting, I think it will be useful to identify the kind of data, that whether the data is numerical, nominal, ordinal, what kind of data it is. First, identify what are their what dependent variables, independent variables, and whether these, and then whether the statistical tools applied are appropriate to the kind of data that they've generated. So these will, even if you don't have time to present all these in this half an hour, if you make notes of these things, it will be very helpful when you design your study, when you choose statistical tests, when you write up, all these will come in handy. So that is good practice, I think. So yeah, these were the things I wanted to tell. And yeah, that's about it. Rest of the things I think Madam has told. One more thing, Dr. Sushma, I don't see why this could not have been a double-blinded study as well. No, I don't think it would have taken much more to make it double-blinded, right? What do you think?
See, if they want, they could have done the double blinding or
Blinding,
whatever.
no?
I
Yeah.
think they, yeah, I think they didn't want to go ahead and mask even the drug. So it takes a lot of time for them
Time.
to take out the tablet and put it inside a cover and then give it to them and things like that. So that is
Right.
because of, yeah, all the resources required. People don't
Required.
go for
Uh,
yeah, blinding.
yeah, I just wanted
And
to
then
highlight
as
that there are some. Sorry. Yes, yes.
no, and that's what as Prashant sir said. If we follow the consort checklist, no, we wouldn't have missed the objectives. So that is the reason why after every slide the consort question should come up. Then we will not. If we start answering it, then we don't miss it. And as sir said, in any table, they will give continuous variables where calculate, I mean, compared using t-test and then categorical by chi-square or something like that. Then you can go back and say here t-test was used, here chi-square test was used. So that itself gives you a lot of knowledge when you start writing your synopsis as a PG. So that is, as sir said, is a very, very good practice to identify for which variable what test is used and what kind of variable is that. Okay.
Yeah, that's about it.
A very good evening to Dr. Shushma ma'am, Dr. Prashant sir, and everyone present over here. Well, I just had two queries, if I may say. Is that fine?
Yes.
Okay. So the first thing was regarding the number of patients who dropped out. Well, you told that it was not mentioned in the paper. Actually, it's been mentioned in the paper under flowchart. It has not been emphasized very particularly, but then it's mentioned that 17 patients have dropped out from the study. So that is a mistake, I guess. Then
Okay,
second,
can I add here?
yes, ma'am,
The
yes.
17 people who walked out were before randomization. Okay, so
Okay, oh,
before
yes, ma'am.
randomization, 17 guys didn't want to do, but after randomization, that is the word called as intention to treat. So the intention to treat principle means once randomized, always analyzed. So after randomization, they are claiming all my 90 plus 90 continued with the study. That is where we had the doubt. And then I think you guys have to read up what is intention to treat and what is per protocol analysis. Intention to treat is whenever you have randomized, even my patient goes out of the study after 4th week, I'm going to carry whatever results he had at 4th week as 8th and 12th week also. So that is called as LOCF, I think, last observation carried forward. That is one imputational method, as I told you, how we compensate for missing data. Per protocol analysis means I am going to take up only those patients who have completed the study, who were with me till 12 weeks. So out of 90, if only 70 completed the study, I'm going to analyze only 70 people. So in RCT, what they say is you have to present both intention to treat and per protocol analysis. Sometimes people play with it, the authors play with it, statisticians play with it. Always your per protocol will come out very well. So they present only per protocol analysis and forget intention to treat principle. Intention to treat principle is, as I told you, once randomized, I'm going to, whether the patient is
Hello.
there or not, I will do some sort of imputational method and I'll analyze him. That maintains your randomization. So that's the advantage of intention to treat. So nowadays, all journals are asking you present both intention to treat as well as per protocol analysis and not bias by just giving us. Now there is something called modified intention to treat also. Statisticians will come up with lots of things to say that their drug is very, very nice. So we have to now, you guys have to read a little bit about what is intention to treat principle, what are the advantages, and then what is per protocol analysis, and what could be the advantage or disadvantage of that. Sorry, you had another question. I
Yes,
sort
ma'am.
of intervened.
No, ma'am, it's fine. I actually understood a lot more now. Then the second question was the close envelope randomization, which we were talking about. Actually, Sushma ma'am also answered it before itself regarding the concealed allotment process. But then, can you explain a little bit more about the closed envelope randomization process as such?
Okay, see, randomization, as I told you, you go to any website or you have a table. Sometimes, previously, we didn't have these kind of software. So, alternatively, we started putting in group A, group B, and all that. So, but anyway, whatever is allocated, that should be concealed for the person, for the investigator. That is what it means. Allocation, concealment. There's a little bit of difference between what is blinding and allocation concealment. Once we randomize and we generate the sequence, what we tell usually people is you give off the sequence to a third person. Okay, that is called as, nowadays multi-center trials will have central randomization centers. Okay, so they give it to an agency for randomization. So you, whenever a patient comes in, you see that inclusion criteria is met, then you are going to call up that central randomization center and you say, I have received, I am from this center and I have received a patient. Then they will say, okay, the patient goes to B. Okay, then you are going to put the B treatment will be in a kit, you are going to give the B treatment kit. But in case if I know that if the whole sequence is given to me, 1 to 30 patients, as I told you, first 1, 2, 3, 4 is A, then 5 and 6 is BB, then A, then BB. Then I know A is, it is open label, so I know A is placebo and B is active treatment. So I have all the chance to manipulate. If I think that one patient who looks like, he may not continue with the things, with the treatment and all that, I'll sort of try to put him in placebo group. Okay, I will tell him, you just sit here for half an hour or 1 hour, then I'll see, put you to the trial, then I'll see one more young and educated person or something like that coming, then I'll give him the active treatment, then I'll call this fellow and give him the placebo treatment. So investigator can manipulate. That is why we want the whole sequence to be concealed. There are a lot of other methodologies how you conceal the allocation. Okay, I think if you read it, you may understand it a little
Yes.
more better.
Ma'am, could you repeat that once analyzed motto for the
The
intention to treat?
intention to treat. So see, that is when intention to treat principle is once randomized, always analyzed. Okay, so once I take a patient and randomized him into one A or B. Okay, so whether he continues the treatment or doesn't continue the treatment, whatever happens, for me he is in that group itself. Okay, so one patient completed till 4th week and he left, he dropped out. But what I'm going to do is I'm going to keep whatever scores I got for the 4th week and continue till to 8th and 12th week. So that is one method of imputation. But I think me and Rohan read another NEJM article which was placebo controlled on prucalopride only. But they gave up. They gave how imputational method was done was they calculated the mean for the placebo group, and then they used that mean to fill in all the gaps for the patients who didn't continue, so that randomization is proper, and that is called as intention to treat principle, that always we analyze all the patients and somehow I make up that value what I was supposed to get out of that patient. Okay, so you may feel that if the patient is only not there, how are we going to do all that? But that is how your randomization is done. If you leave all the patients who are leaving and just take up 50 patients in placebo and 70 patients in prucalopride group, then your randomization itself is sacrificed. So there is no point in saying that it is a randomization study if you take per protocol analysis. So always analyze everything, but you have to give us in methodology how you are going to make up that missing data, and it has to be scientific. So they are called as imputational methods. If you read about it, there are lots of different ways about how to do imputation. Too many imputational methods are there. They can select any one to be used for intention to treat principle. Okay, I hope I am.
So
No,
much.
you guys are. It is all little too much, I think.
Yes, ma'am, it is. But I think we'll all get to read about it. You've given us a
Yes.
lot to
Yeah. I
read
think.
about.
Yeah. Once you start reading, I think you guys will
And
start
at
understanding.
a UG level, we have not been exposed to randomized control trials as such. We just
Yeah.
read
Yeah.
normal articles, review articles. That's
Yeah.
why
Yeah.
this
Yeah.
is
Yeah.
very interesting.
That's true. That's true. Yeah. 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 You get to see and do journal clubs as PGs at that time. I think even we took quite a lot of time to read, but I'm very happy that you guys are getting exposed to all such kind of things right now itself.
Thank you, ma'am. Anybody have anything else to add?
Yeah, I asked a few more faculty to join. I think Aradhna joined from orthopedics. She might have left. I don't know.
Aradhana, ma'am,
Aradhana
are you
and
there?
Preetham, sir.
Hi, ma'am. I have joined.
Oh, thank you, Aradhana. Thank you. You also can give us some
Yeah.
comments.
Oh no, ma'am. This is like these people are presenting. Even I gained so much knowledge after Rohan presented. It was a very good presentation, ma'am. Very good. In
Thank you,
his
ma'am.
age, we didn't know what RCT was. We had no clue. So it's good that they are exposed to these things which are really necessary for progress. It's very good. It's nice, ma'am. It's nice.
Thank you so much, Aradhana. I think you could, Manin, you can just check. Some of the faculty might be there. I don't know. Or you have to switch on your mic and say that you guys are there.
Yeah, true, true.
Preetham,
Thank you
sir?
so much, Aradhana.
My pleasure, ma'am. It was good. I learned so many things.
Oh, thank you so much. Thank you so much. And a big thank you to Rohan, actually. And he really worked very, very hard for this presentation.
Thank you, ma'am.
Yeah. Thank you, Aradhana, ma'am, as well, for joining us.
If no one has questions, can we just close down, Mani?
Yes, ma'am. That's totally fine. Okay, then let's end the meeting. Thank you everyone for joining. I hope this was a useful session. And once again, congratulations, Rohan, for such a wonderful presentation.
Thank you, ma'am.
Good night.
Okay,
then.
thank you everyone. Good night.